This is our last set of notes on evolution, and this is Patterns of Evolution in Early Earth History. So making connections, we have Big Idea 1, which we're still in. Speciation and extinction have occurred throughout Earth's history. And then we have those changing conditions, which are going to change homeostasis and development, which is Big Idea 2. So let's start with Earth's age and formation. Most scientists will agree that Earth started forming about 4.6 billion years ago, that's the BYA, from a condensing cloud of dust and rock that was spinning around the sun. It isn't until about 3.5 billion years ago, so we've got about 1.1 billion year time span where no life was on Earth, that we actually start to see the first fossil prokaryotes. Now remember, a prokaryote is an organism that is single-celled, does not have a nucleus, and doesn't have any other membrane-bound organelles like mitochondria or chloroplasts or Golgi bodies. And then we don't get to see the first eukaryotes, those having a nucleus, until about 1.8 billion years ago. So you could say that those bacteria were alone on the planet for quite a while, almost 1.5 billion years. The conditions of Earth's early atmosphere were very low oxygen, lots of toxic compounds that were being released by volcanic eruptions. It was a very hostile place, most scientists predict, as the planet was developing and things were coming together. The landforms were being made, the oceans were being condensed and formed. We probably didn't have water for at least a billion or close to a billion years. Then once we had water, there were all kinds of things in there: carbon, nitrogen, sulfur, phosphorus, and those came together to make what we now know as life. So O'Parent and Haldane, they were two scientists who separately hypothesized that our atmosphere was one that we would call reducing, which means it was adding electrons to it, made it a very charged environment, and the oceans became this soup of organic molecules, like carbon with hydrogens on it, and nitrogen with oxygen, and sulfur oxides, and all kinds of other ingredients that were in this quote-unquote soup. That's the primordial soup or primordial ooze that people refer to when they say, like, we came from the primordial ooze. So that's what they proposed. Miller and Urey were two scientists who actually tried to test their hypothesis, and they did this by making comparative lab conditions. Now, O'Parent and Haldane said that the energy for all of this came from UV radiation and lightning. So they used electricity and some other components with some of those organic molecules in a water-based soup, if you will. And they actually hit it with the electricity, and what happened was they yielded amino acids. So from their experiment, they said that amino acids and other macromolecules can form from smaller organic molecules if the conditions are right. So it didn't prove the hypothesis, it just gave us a way to say, okay, this could have been true, this could have happened. In these conditions in the laboratory, we were able to create macromolecules. So this is what they thought happened. It was basically a 4-stage process that our Earth had to go through in order to produce cells, and those first prokaryotic cells about 3.5 billion years ago. So the first part of the process was that we had to have small organic molecules like the CO2 and H2Os, and well, H2O is not organic, but everything with carbon in it, CH4. And we had these small organic molecules. Those joined together to make larger macromolecules, which then self-assembled into packages that scientists called protocells. So not quite a cell, but they did have this membrane that separated them from the rest of their environment. And if you remember from our chemistry lesson, if you want to remember our chemistry lessons, a phospholipid bilayer, if put in water, if you have those phospholipid heads and the fatty acid tails, it will actually form into that layer of protection. So that's where they get that from. And then from the protocells, they must have had some sort of RNA, which they think was the first, which we'll talk about in a second. And then they were able to make self-replicating molecules. So that first protocell had to be able to make another protocell, and then another protocell, and so on and so forth, until we actually had full cells and then a fully self-replicating organism. So that would be asexual reproduction. Okay, the first life, again, we said we're prokaryotic cells. Protocells would have been first, but prokaryotic cells would have been the first, like, real life, because we define life as having a cell. Most likely that life contained RNA and not DNA. DNA, remember, is a more complex structure. It's two-stranded and has deoxyribose as a sugar instead of just ribose. So RNA is a pretty simple structure. And then also, if you remember from biology, RNA is the one that actually assembles the amino acids into proteins and with those peptide bonds. So it makes sense that RNA would have been the first thing to be around, and then DNA would have been created as a storage molecule, a larger storage molecule later on. That first life were the prokaryotes, and then came the first eukaryotes, the ones that had a nucleus, but they were unicellular eukaryotes. We would know these today as the protists. So the unicellular eukaryotes were formed by something that we think is called the endosymbiont theory. I'm going to show you a slide of that in just a second. But right over here are the different types of bacteria. Now, these are just three shapes. There are many different kinds of bacteria. Okay, so now for the endosymbiont theory. We have this cell up here, the ancestral prokaryote. You'll notice that the DNA is not contained in the nucleus, and you cannot see any organelles floating around in that cytoplasm. Well, what scientists believe happened was there was a folding inward of that membrane, and that created what we know as the endoplasmic reticulum and also the nuclear envelope that helped to surround the DNA. So that's how we got the nucleus. This cell actually took in an aerobic bacterium, so one that was using oxygen to create a food source for itself. So this one takes in this bacteria, and that bacteria started to live inside. Endo meaning inside, symbiosis is a joining together to work in either harmony or discord, but joining together to work. So we have the mitochondria inside of the cell. Actually, it was just a bacteria. It became the mitochondria. And then if that cell also would have engulfed a photosynthetic bacterium, like in algae, then we would have had a plant cell or a eukaryotic cell that had plastids or chloroplasts and other organelles. So we started with just the prokaryotic cell up here, and then made our way all the way down to animal-like cells here and plant-like cells there. So based on biochemical analysis, these are the origins of what we now think are key eukaryotic features, like the apparatus that helps in cell division or the mitochondria. And over here is the source. Now remember, there's two domains of bacteria, the archaea and the bacteria. And so we have the archaea, which are the like extremophiles. They live in crazy environments, organisms like that. And then the bacteria, which live pretty much everywhere else and have slightly different cell wall structures and things like that. But that's where we think we got the eukaryotes from, or the eukaryotic parts from. Okay, so looking at this picture, where would you guess that these dinosaurs were roaming? Would you assume that it would be the Antarctic? I didn't, and I think that would surprise you too. We see the trees and the life, and now the Antarctic is a lifeless place where there is nothing smaller, or nothing, excuse me, bigger than a few millimeter bacterium or algae that could possibly live there. But what our fossil record reveals to us is that our Earth has been an ever-changing and vastly changing place. If this is what Antarctic looked like millions of years ago, what did the rest of our Earth look like? This is a picture of Tiktaalik, which if you read Your Inner Fish, then you'll remember the significance of this organism. It's an extinct aquatic organism that is the closest known, as of right now, relative of us, the four-legged vertebrates. Well, not directly us, but the things that gave rise to us. And they were the first four-legged vertebrates that went on to colonize land, and then, of course, gave rise to us. And there's all kinds of really cool stuff in that book, if you get a chance to read it, about this fossil and the discoveries they made. But that's just one part of our fossil history and documentation of life on this Earth. Okay, so here's another timeline that shows us, like down here, 3.5 billion years ago, we've got the fossil prokaryotes. They're called the stromatolites, which were actually layers, and then we can see the fossils in them. We've got this, which was thought to be like a fossilized, either a fungus or an algae. Working our way up, we can start to see structures forming. Okay. And then in the millions of years ago, we have the dinosaurs and our relatives. The fossil record does show us that there's been a lot of change in Earth's history, but it's often argued that the fossil record is incomplete, and that is totally true. There aren't very many organisms or parts of organisms that can actually be fossilized. If you think about the fossils that we do have, they're bone or shells. So any soft organism was most likely not fossilized. We do have a few impression fossils of those organisms, but those are few and far between, and actually very rare. So we don't even have a full picture of what the diversity of life on our Earth really was. It could also be that the organism just wasn't in the right place at the right time with the right conditions to be fossilized, even if it did have bone or shell. Every day, though, gaps in the fossil record are being filled. And I just put that cartoon because I thought it was funny with the large organism trying to, whale-like organism trying to walk on 4 legs and then get into the water, and it says it's all yours. Then you got this little fishy guy coming out of the water, just to be kind of silly. So how do we know the age of the fossils and the things that we're pulling out of the earth and studying, and we're trying to determine how old the earth is and how old these organisms are? We have 2 different methods. One is the relative age, and that we look at the rock layers. Which rock layer came first is probably the one that's on the bottom, and the one that's on the top is the one that was most recently laid. So it would be kind of like peeling off wallpaper in an old house. You can infer that the first layer of wallpaper you pull off was the most recent to be put on the wall. And then when you keep getting down and down and down and you get to the actual wall itself, then you know that that had to be put in first or laid down first. So the other thing that we can do is absolute dating, which doesn't mean it's perfect. It doesn't mean that it's like 100%, and it doesn't give us like this happened in December 14th of the year 65 million BC. Okay, but it does give us a date and a time in millions of years ago or thousands of years ago that we can use to sequence events in Earth's history. One of the ways we do this is through radiometric dating. This is where we take isotopes that we know, for instance, carbon-14, which decays to nitrogen-14, and we know that carbon has a half-life of 5,730 years. So every 5,730 years, half of the carbon-14 will be left from the original sample. We know that these isotopes and their half-lives are not affected by temperature, pressure, or other environmental variables. Why is this important? Because then we can say that, no, this wasn't just because these conditions were happening at this time and now we have these conditions. We can say that these things, these isotopes decay at this rate no matter what. Okay. So how do they use this? They look at the amount of carbon-12 that is in the organism, and that amount does not decay. Carbon-12 does not decay. It's not radioactive. So they can compare the amount of carbon-12 with the amount of carbon-14 that is left in the sample, and then they can know the age from that. So they take the carbon-12, find out how much of that there is, find out how much carbon-14 is left, and then they can see, okay, this much carbon-14 has decayed, so that means it's been this many years. The only problem is this only works up to about 75,000 years to actually look at the sample itself. Another way they can do this is to look at volcanic rock, because as the volcanic rock is cooled, it traps isotopes that are not common in organisms, things like uranium-238 or other forms of isotopes that maybe won't decay as quickly. And we can actually take a look at organisms that are in between 2 of the igneous rock layers, and then we can determine, okay, this happened, this rock was created 100 million years ago, this rock was created 125 million years ago, so this fossil is somewhere in between that age. Here's just a graph showing the half-lives and the amount of parent isotope that remains. From all of our studies, we've created a geological record, which is basically a standard time scale that's going to divide Earth's history into 4 eons, and we'll take a look at those. Pull this up here. So the Hadean, the Archean, the Proterozoic, and the most recent Phanerozoic. And then each of those eons will be divided into eras and periods and epochs. And thankfully, we don't really need to know any of this stuff. We just need to be able to recognize that the Phanerozoic is the most recent, and that's our age, and that most of the organisms that we know have come in that eon. Okay. Here's a picture, cross section, and then also what those stromatolites look like. We can use our dating methods to determine that these were about 3.5 billion years old. We can look at organisms like this and look at our geological time record. We can actually build our geological time record and then use that to place organisms and to try to determine what developed, which organisms developed from which ones, and the relative timeline of their development, the structures that developed, and everything like that. So here we have a diagram of what would be the origin of mammals, and these would be fossils that show morphological features that would be intermediate steps between the synapsids, which were about 300 million years ago, to the modern day tetrapods, our relatives and mammal relatives. So you can follow the diagram and see the changes in the bone structure, changes in the hinge of the jaw, changes in the temporal region as the organism developed. So that's another example of how we can use different groups of organisms that have been around and then are extinct to build our geological time, and also to see extinction and speciation rates. We know that species diversity is going to increase when there's more species that originate than are lost to extinction. So here we have a hypothesis that by about 2 million years ago, both the lineages of A and B had given rise to 4 species. So if you can follow the 4 different species of 2 million years ago. So we're right here. 1, 2, 3, 4. If we keep following the lineages, we notice here the daggers mean extinction. That lineage A, out of all the organisms that it produced, almost all of them went extinct except for that one up there. But in lineage B, by the time we get to present day, these organisms are still around. All of these that don't have daggers with them, and this is the only one that went extinct. So the lineage B had a greater speciation rate and was able to affect diversity more. Another thing that affected the amount of organisms and where they lived and how they developed was the Earth itself and the movement of the Earth, known as plate tectonics. So we know that the continents are part of great plates on the Earth's crust that are floating on the mantle, right in here, right? And the continents would be up here on the crust, and they're floating around on there. Floating is kind of a weird term, but they're on there and they're moving, okay? And we know that we can measure the rate of movement, and we actually know how long ago the continents were in different positions. So here are those plates, and we can take a look at the arrows, which are indicating the direction of the movement of those plates. And then we can also see all of the red dots, which would be zones where we have violent tectonic activity. So right along the Juan de Fuco plate, over here, there's going to be lots of earthquakes and activity. So everywhere you see those red dots, so we know that those plates are moving, and we're not going to get into the whether they're, slipping under each other or going over each other, but we know that Earth structures were formed this way. Mountains, ridges, and valleys, and all kinds of other geographical processes were taking place. Islands were forming. Okay, two plates were moving, could be moving away from each other, and we've got this situation where they all are now and where the continents are, and that's going to determine the diversity and the types of species that can live in those areas. So we have this ever-changing Earth that we see. So here is another graphic just illustrating the change. Like 251 million years ago, we had Pangea, the supercontinent. So you can see where we would have had lots of organisms in one area, and it was all pretty much the same type of climate. But then by the time we get up here to about, well, the positions that we're in now, about 45 million years ago to present, we know that the environment down here is way different from the environment right here along the equator. So different kinds of organisms are going to be there. So it's just another way of just saying that our Earth's history will dictate what kinds of organisms can be in which places at which times. So we're going to talk about extinctions and how those affect the diversity on our planet. So see if you can pick out where the they call them the big five extinctions, or mass extinctions, excuse me, happened in our history. Go ahead and pause the sound if you need to. Okay, so did you find them? We've got one right there. A little bit smaller one, two right there, huge one right there. Number of families was 1.1 million families. Another one there, and the fifth one here. Okay, so where were these different extinction events and what was happening? And you can see that then the rest of the graph, we've got pretty low levels, or relatively low levels of extinction. So let's kind of talk about those big five. A mass extinction, obviously, we're losing lots of organisms. It's going to be a disruptive change to the diversity, and it's usually caused by a disruptive change. So something happens in the environment that's going to cause these mass extinctions, which then cause a disruption in the diversity of species and in just sheer numbers of species, which will change what types of species are available and the environments that they're available to live in. There have been those 5 mass extinctions over about 500 million years. We can see that from the fossil record, where we'll have organisms and then nothing, and then all new organisms that we'll see fossilized. So during those mass extinctions, about 50% or more of marine species become extinct, except in the big Permian extinction that happened. It's the separation between the Paleozoic and Mesozoic eras. If you need to go back to that previous chart, you can see the difference, the Paleozoic and the Mesozoic. That was the one where it had 1,100 million different species went extinct about 250 million years ago. We lost, estimates are about 96% of the marine life during that extinction. So we need to ask ourselves, with the way our planet is headed and the things that we do to it, will we be here during number 6, and will we be the cause of the number 6 mass extinction? There are many scientists that believe that we are well on our way to causing the big 6, where we will lose millions and millions of species. In just the last 400 years, more than 1,000 species have become extinct from human activities, which is about, depending on estimates, 100 to 1,000 times the normal rate of extinctions. And we do know that warmer temperatures do equal more extinctions. We can see in our graph here that we've got 4 of the big mass extinctions happening during warmer temperatures than during cooler temperatures. So the consequences of these mass extinctions, okay, yeah, we lost all these organisms, the diversity of life has gone down, but there are long-term effects. Mass extinctions totally transform ecosystems into shadows of their former self. Sometimes, and most times, it takes, based on the fossil record, 5 to 10 million years or longer for nature to recover from a mass extinction. I don't know about you, but I haven't been here that long, and so if there were a mass extinction, we definitely would not be around to be able to see the organisms that come out of it. It's not going to be a simple and quick fix. What a mass extinction does, because it transforms the ecosystem, is it changes the types of organisms that are going to be successful and be around in that new ecosystem, and it paves the way for adaptive radiation to happen. During those periods of evolutionary change, many new species will be formed that have many different roles. The Permian extinction took over 100 million years, according to the fossil record, to recover. Depending on the species that are available, more or less predators, will determine what species will survive and make more and newer species. Here's an example of the adaptive radiation of mammals, where we have our ancestral mammal here, and then lots of new species being formed there. So that's a period of evolutionary change right in here. Got 2 periods of evolutionary change where groups of organisms form many new species. The Hawaiian Islands are also another great example of adaptive radiation. All of these organisms that you see here are relatives of the silver sword. They're part of the Silver Sword Alliance. That sounds cooler than it actually is. They're just different kinds of plants that all radiated from one ancestral species that was brought to the islands. Not really sure how, but it was brought there, estimates about 5 million years ago. And now we have all of these different kinds of species. So just like the graph that we just looked at with the mammals, where we have an ancestral mammal making all of the monotremes, the marsupials, and the eutherians. Now we've got an ancestral plant making all kinds of different plants that genetic analysis can tell us are actually very similar to each other. Now we're going to talk about the effect of developmental genes on evolution and changes in body form. So the first thing that we're going to talk about is why I typed effect wrong. No, we'll cross that out. So it should be effect, not effect. So there's changes in rates and timing of developmental genes, and that's going to determine the morphology or the physical look of an organism. Okay, so the organism's shape can depend on the growth rate of the body parts during development, which is an easy way to say that. So here we have a table or diagram that shows the chimpanzee infant versus the human infant. So you can see the skull development and how quickly the chimpanzee will be shaped into this like oblong shape of their skull, whereas the human development has the more characteristic, almost like a spider abdomen look to the skull. So those are developmental genes and the timing of those that are causing the differences, because the bones are the same. It's just how they're shaped and formed that's different. Another thing that we can look at over here is the bat, hand, and fingers. So they have the same bones as other mammals, but because of the timing and the genes that are involved and the rate of development, this bat will have very long, slender hands and fingers versus another mammal. Another timing thing that we can talk about is Paedomorphosis, excuse me, that's a mouthful. And this is where we have a sexually mature organism, so one that is able to have babies, but it's going to retain juvenile structures like this salamander here. So you can see that it still has the juvenile gills, but it is actually a fully formed adult capable of having offspring. There are genes that actually control the placement and organization of body parts. This was also in the book Your Inner Fish. So some of you that read that book all of this is coming back, and it's, actually an application of what you got to read this summer. But these homeotic genes, they're going to control the placement and organization of body parts. So for instance, where do wings or legs develop? And if you read the book, you remember that they were able to mess with some of these and cause wings to develop on a head or legs to develop in a different place than what they should. One of those types of genes were the Hox genes, and those determined positional info inside of the embryo, and they were able to cause really crazy cool things to happen, like doubling the digits. Okay, so it caused the digits to develop both on the left side and the right side. Novel forms, body forms, are thought to have arisen from events like this in those Hox genes and other genes where there's been a duplication event that's caused the organism to have more legs or more genes, which will either be turned on or off, which will create more structures or less structures, like in the fly here and this like primitive shrimp-like organism, the Artemia. So this one has lots and lots of legs, but the UBX gene is not turned on in the Drosophila, which causes the fly not to have all of these legs in that same place. So still has the gene, but it's not functional. There are changes in gene regulation too that affect how and where other body parts develop. So based on this study here, we'll zoom in on this. Okay, this is the stickleback embryo, and they were actually able to mess with the genes of the stickleback embryo, and they hypothesized, was this a result in a difference in sequence, or was it a difference in the expression of the genes? So is it that we changed from ATC to AGC, or was it that they have the same sequence, but they don't have the same expression of it? So in hypothesis A, there was no result. All of the amino acids are exactly the same. So then they concluded that the difference was in the expression that was creating these, they're right here, these ventral spines, you could imagine that if a predator is after you and they go to bite you and you've got these spines here on your belly, that's probably not going to feel too good. In organisms that lack those spines, they don't have predators in their environment. So it's just a difference in expression of those genes that causes the two different organisms. We have to remember, because sometimes when we talk about this stuff and it seems like we're saying that these things happen on purpose, or evolution caused these things to happen on purpose. But we have to remember that evolution really has no goal. It just works with and edits whatever is currently available based on the environment and the time. So an organism doesn't think, oh, that would be great to have this, and then it develops it. That doesn't happen. And remember, evolution can only work on populations, not on individuals. So where do we get these novelties, these crazy complex structures that we see? One of those complex structures we actually read an article about in the Evolution of the Eye. And the question is, can these complex structures evolve in gradual increments? What would be the benefit to this of an eye that doesn't fully see? So if you remember reading in our article, with the human eye, there are other eyes in other organisms that aren't as fully developed as our eye, and they do serve functions for that organism. So it is reasonable to think that through many different editing and mutations, our eye could have evolved in a gradual increment. So finally, our last topic is on trends. Can we pick out evolutionary trends in lineages? And yes, we can. We can see that some lineages have a tendency or a trend towards getting larger, or some have a tendency towards getting smaller in their body types or in their structures. Just like a population can undergo selection and have evolution from that, we can say that species can undergo a type of selection called species selection. And this is the species selection model. If we look at this, speciation events are the birth of the species. Extinction would be the death of the species. And then new species that are created from that species would be their offspring. That's a lot of species in a row. Hopefully you understood that. So the species that produces the most new offspring, quote unquote, species, will determine what the trend is. The more offspring that species generates, the more the trend will go in the direction that species lineage is. So let's take a look at an example. Okay, in our final slide, here is the horse evolution from about 55 million years ago. We have this almost dog-sized horse. Okay, that was the species. If we take a look at the next one up here, a little bit larger, a little bit larger, larger still, and then finally the modern horse is very large, very different body structure compared to that little dog-like horse of 55 million years ago. So as the evolution occurred, new species were formed, speciation occurred, there was an evolutionary trend to grow bigger and better body parts. Could this trend have changed? Certainly, at any time. An environmental condition could have caused the trend to go back down, and it still could, or it could have caused it to go in a totally different direction or just stop progression altogether. So An evolutionary trend is just kind of another one of those relative things depending on what we're looking at and the environment and the lineage and how many new species are being created. If you have any questions, let me know, and I'm glad we got through all 7 sections of our evolution notes. See you in class.